Welcome to our fourth fully vodcasted lecture. This will be our final fully vodcasted lecture unless we decide we really like the idea of trying the upside down classroom. And then we may do another fully vodcasted lecture for fun where we would do the vodcast as the lecture, and then during lecture time we would simply review and talk about homework and talk about the concepts. So that upside down classroom idea is still out there and on the table, but this is the last time that I will be gone for a Friday lecture. I wanted to make a few announcements regarding exam number 3. I hope everybody knows that they can find the review under doc sharing on eCompanion, and maybe you've already made some good use of that review. And of course I'll be handling in-person reviews as well from 3 to 4 p.m. hopefully in the classroom itself, but if not, upstairs in the lab, and also on Illuminate at night at 7:30 to 8:30 p.m. on Monday. So I'll look forward to reviewing with all of you, and hopefully the poster making has been just going gangbusters and you're feeling really good about the upcoming exam. In terms of the poster presentation, I wanted to make a couple of announcements on that. I got some questions about the general nature of the poster. I want to make a quick review announcement that you can find the vodcast on iTunes U that talks orally about the poster presentation. Of course, the assignment description is also on the syllabus that I passed out on the first day, and I'll show you in just a moment where it can be found on the web page as well. I want to also mention for lab that you're starting to get to the point where you may be thinking about what your unknown microorganism is, and as you begin to think about that, make good use of the online features, the dichotomous keys and the tables that are online, and I'll show you also where those are. So without further ado, let me pull over the lab website so that everybody can take a quick look at those the assignment description for the poster, and also for the lab where you're going to find some of the additional information for your dichotomous keys and your tables. So let's start with the latter, looking at the additional information on the lab website, and under that additional information tab you can actually find here the dichotomous keys, and these are down under Material to Accompany Labs 15 through 28. So you can click on the dichotomous keys. The printable version is largely what you'll want there. You're probably also going to want the tables that are available, unknown ID tables. These are going to be super helpful because whereas the dichotomous keys are one person's way of putting together these data, the tables include everything. So you're probably going to want to grab both of these as you begin to think about how to identify your unknown. These will be great resources and something that will really help you as you get down to the nitty-gritty on the biochemical features of each one of your unknowns. Let's take a moment to pop back out here and look at the lecture website where under additional information you can also find the current events assignment description. And remember to take a look at that vodcast that's on iTunes U that takes you orally through all of that and gives a description of the assignment, making sure that you're really taking use of that. And of course right now you're starting to think about submitting those abstracts, and so the abstracts will be due after the exam. I've decided to kind of postpone that. I know we're not quite at the point where everyone can submit those. So when you do sit down to make your abstract, make sure you run down here in the assignment description and find the section that is regarding the abstract. And that section is just down here. And you're going to be able to find under this section how to write an abstract. So an abstract is a 1-paragraph summary, about 200 to 300 words, and it tells you exactly what's in it. Here's the general structure of an abstract. So use that to your advantage when writing your abstract with your poster groups. I hope you're having a super good time, and it seems like the interaction has been way awesome, especially after I made that Harry Potter analogy. So keep up the great work, and let's dive in because we have some very exciting things to talk about. After having wrapped our minds around transformation and the uptake of naked DNA, we now want to turn our attention to transduction and the idea that DNA can be transferred from a donor to a recipient via a taxicab bacteriophage. So we're going to talk first about the 2 different types of bacteriophage, reviewing from lecture so that we feel ready to go into the discussion of transduction. I want to make sure that everybody is recalling the 2 types of bacteriophage that we talked about in lab. So we had 2. One of them we focused on One of them that I joked is sort of the quote-unquote stupid type of bacteriophage. That particular bacteriophage is the one that gets in, replicates, lyses the host cell, and gets the heck out of Dodge. So it really burns through its host cell. And in that way, we kind of think of it as being sort of stupid because of course, a bacteriophage can only replicate within its host cell. And so if it's burning through its host cells, then it's not leaving a whole lot of options for itself to replicate. However, many bacteriophages do successfully use this strategy, obviously. The T4 bacteriophage is one of those. So these are called virulent bacteriophage, and they follow the lytic pathway. I imagine a lot of you are remembering that pathway, and in a moment we will review it further as we think about it in the context of transduction. The other type of bacteriophage that we talked about is perhaps the more smooth of the criminals. This bacteriophage is called a temperate bacteriophage. Temperate bacteriophage can follow either the lytic pathway or another pathway called the lysogenic pathway. And the lysogenic pathway is where these are pretty smooth. In the lysogenic pathway, what happens is that these bacteriophage actually integrate their phage DNA into the host cell DNA. So let's actually jot down a quick picture of this where we have our bacteriophage. We'll try to get a reasonably good picture of a phage with its icosahedral head here. And it has landed on the surface of a host cell. And into that cell, penetration has occurred, and we get the introduction of 2 strands of phage DNA. We're going to follow it that way. And remember, of course, that we have the bacterial chromosomal DNA. And in the lysogenic pathway, what will occur is that that phage DNA is going to integrate into the host cell chromosome. So we're going to get a situation like this, where in that host cell, the phage DNA has, like a smooth criminal, has integrated into the host chromosome. So that each time that the host chromosome is replicated, so too is the phage DNA. Pretty dang smooth. So we're going to recognize this as being a pathway that's followed by a temperate bacteriophage until it feels a threat. And if it starts to feel a threat, that's when it's going to excise its phage DNA from the chromosome and go right into that lytic pathway and get the heck out of Dodge, right? So this is a pretty quote-unquote smart bacteriophage. So keep this in mind as we go into the next section where we will talk about transduction, where in transduction, this is a means of bacterial gene transfer via a phage taxicab, where the phage is picking up some of the bacterial DNA and taking that bacterial DNA from a donor cell to a recipient cell. This can happen with both temperate bacteriophage and virulent bacteriophage. The type of transduction that occurs, there's one type that can only happen with the lysogenic pathway, and then there's another type that happens within the lytic pathway and is much more broad. Let's go ahead and review those 2 types of transduction. So after a phage infects the host cell, multiplies, and is released, it may come out being what we call defective, meaning that rather than incorporating just its own phage DNA, it accidentally incorporates some bacterial genes in place of those phage genes. Now, this can occur in in the lytic pathway, remember how when a phage gets in, it undergoes penetration, it undergoes biosynthesis and maturation. Well, remember that during biosynthesis, the hijacking of the host cell, there's a lot of degradation of the host chromosomal DNA. So there's a lot of parts is parts, pieces is pieces, pieces are pieces of the chromosomal DNA of the host. So you can see how it would be easy for a phage to go wrong and inadvertently package DNA from the host into its capsid in place of its own DNA. So phage-containing bacterial genes now are found in this phage, and it goes off thinking it's going to head off to another host, infect that host, and be able to cause, to replicate its own DNA, but in fact, it only contains bacterial DNA, or it contains partial viral DNA, so it's unable to successfully do so. But what it does do is it transfers genes from the first bacterium to the second. So these two types of transduction, as promised, the more generalized transduction, in which during the lytic pathway, the phage incorporates any random piece of DNA that happened to be there in that parts is parts sort of sort of geography of the cell at that time. Versus specialized transduction, this is the type that can only happen with the lysogenic pathway, where the DNA has integrated into the host chromosome. Now, this is when that DNA has incorporated into the host chromosome, it's called a prophage. So specialized transduction can only happen when a prophage has been formed where it has integrated into the host cell chromosome. So in generalized transduction, during the maturation phase of the lytic cycle, the phage mistakenly packages bacterial DNA, and this can be any old random gene. There is no predicting which gene it will be because there's so many random genes hanging out and they've all been degraded by the phage infection, and so in fact any gene could get incorporated. Now maybe you're thinking, well, gosh, I wonder if the whole bacterial chromosome ever gets packaged into this phage taxicab. I mean, does it ever just like pick up the entirety of the entire, genome of the bacterium and transfer this whole genome to the next host? Well, this is impossible because remember how tiny bacteriophage are. They're on the order of 100 to 1,000 times smaller than the cells that they infect. So that being the case, it can't package that much DNA into its capsid head. And in fact, it generally tends to range from about 1 to 2.5% of the chromosome of the bacterial cell that can actually get packaged into there. So it's a very tiny percent, and of course that depends upon the size of the capsid of the particular phage. Now, in specialized transduction, things are a little different. I'm going to come back to generalized transduction and show you some pictures. But in specialized transduction, what happens is, remember how we mentioned in the last slide that when a bacteriophage feels a threat and it has integrated into the chromosome, it will excise from that chromosome and it will go into the lytic pathway and get the heck out of there. But sometimes what happens is when it goes to excise, it makes a mistake, an error. And instead of excising properly and just taking phage DNA, it actually takes some of the neighboring or flanking bacterial DNA with it when it excises. So the phage is, basically defective, but on the other hand, it has now picked up some bacterial DNA that now is packaged into the capsid and can get taken to the next host. So an error is made during the lysogenic life cycle, allowing the phage to take some of the host DNA when it leaves the host chromosome, about 5 to 10% of the flanking DNA. In this type of transduction, only a few genes next to the point of integration may be transferred. So that's the nature of specialized transduction. And in your textbook, if you're just feeling extra sexy minded, you might want to go in there and check it out. There's a little bit more, I should say a lot more detail on specialized transduction. For our purposes, let's look at the details behind generalized transduction at this point, looking at an image where we can see what happens after this bacteriophage takes its DNA and heads off to a new host. So let's begin with the process by which that defective bacteriophage is made, and then end with what it does with the host DNA as it goes to a new bacterial cell, This picture should look awfully familiar to you. We've got our stages, right? Remember our all people buy moldy lemons. This is beyond the adsorption phase where now everything has attached, and then during penetration the phage ghosts are made after the DNA from the phage leaves and heads into the host. Once inside the host, those early and late viral genes are expressed, leading to the degradation of the host cell chromosome. You can see there the red DNA has been degraded, and of course leading to the expression of viral genes, both those involved with genome replication and in biosynthesis of all of the parts of the tail plate, the base plate, the tail fibers, the sheath, the core, and all of those components of the bacteriophage. But see how messy it is in here during biosynthesis, and how there's a lot of viral DNA, but there's also some of this bacterial DNA that was degraded. Well, this bacterial DNA can get packaged inadvertently into one of the phages, and we can pick that phage out here, and amongst all of these daughter phages, we can recognize that the one that is quote-unquote defective is this one here, because it is packaged red DNA instead of the black viral DNA. It's now carrying the red DNA of the host. So this is the one that is defective. That's one term for this bacteriophage. Another term that it is sometimes called, if you're looking for the more technical term, is a generalized transducing particle. So that's the tongue twister name for this bacteriophage that now carries the DNA of the host bacterial cell rather than having its own viral DNA. So if you want to jot down on there that not only is this known as the defective bacteriophage, but it can also go by the name of generalized transducing particle, because this is generalized transduction, and this is the particle that's making it possible, right? So generalized transducing particle. Very sexy term. Okay, very good. So now we want to follow the journey of this defective bacteriophage. As it heads out and does what phage do, it finds a new host, it adsorbs to that new host, it then undergoes penetration and all of the early steps in the lytic cycle. But of course it carries with it the DNA of a bacterial cell and can't hijack the host cell, because all it's bringing in is some new viral or some new bacterial DNA. So taking a look at what happens at this point, penetration has occurred, it introduces that new DNA, but of course that's bacterial DNA, and now a couple of different fates can occur with that newly introduced bacterial DNA. One of the things that can occur is what we call complete transduction, and that's actually what we're seeing in this first image on the left, where the newly introduced bacterial DNA that came from the virus is going to integrate into the host cell chromosome. So this is what you would think of as complete transduction. I'm going to jot that down. Complete transduction. Where effectively now the donor DNA has integrated into the recipient chromosome. And of course now Claire will also be remembering that we would now accurately call this cell a recombinant, right? Recombination has occurred. So the type of lateral gene transfer that has occurred is transduction. The cell now is a stable recombinant because complete transduction has occurred and the donor DNA has gotten integrated into the recipient cell's chromosome. But what's interesting is that a vast minority actually of the DNA is actually stably incorporated and integrated like this into the chromosome. And in fact, about 70 to 90% of the DNA, it's brought into the cell and it actually persists. Now this is weird. You guys are going to think this is so weird, because what happens here is that this DNA persists. It stays in this cell at least for one generation, and it will continue to be expressed. Like if any genes that are on there will be expressed, but it may duplicate some of the genes that are already in the host cell chromosome. So here's where this is crazy. It means that we get, transiently at least, an E. coli host cell that is partially diploid. That is so weird, right? It may have more than one copy of the same gene depending on what came in on that donor DNA. This is called abortive transduction, and as I said, about 70 to 90% of the cases this is going to occur. So this is pretty crazy. Now sometimes this foreign DNA is recognized, and imagine Carl is thinking, oh yeah, but a lot of times doesn't that look like a foreign particle there, and that the host cell says degrade it? And sometimes that is the case, but it's amazing how often we see abortive transduction. So degradation is yet another fate that we might see in this process. So this summarizes our coverage of transduction, and we now find ourselves ready to go into what is my absolute favorite mechanism of lateral or horizontal gene transfer, conjugation.